Hello, and welcome to Let's Talk SADS Live, the Canadian webcast from the Canadian SADS Foundation. I'm your host, Dr. Shu Sanitani. Delighted to be coming to you live from Vancouver, and greetings to wherever you are listening to this, and really excited to have this episode. You'll notice my background's a little bit different. This is a real background coming to you from my home instead of the studio that I usually come from. So we're going to try and do this seamlessly here, even though it's home, and I'm going to share some slides. Okay, so episode 34. Wow, and so glad to be part of this. The Canadian SADS Foundation. SADS, of course, sudden arrhythmic death syndromes, conditions that have impacted many of you with sudden arrhythmia, cardiac death, cardiac arrest. And we are here to learn together collectively and collaboratively about these conditions. And we've learned so much about these conditions in the last decade. It feels like an incredible explosion of knowledge, which is part of why we're excited to be here sharing this with you. We've got an exciting discussion today. So we're here, of course, thanks to many partners, including, of course, the back office at the Canadian SADS Foundation, and of course also the American SADS Foundation, and Alice Lara, who recently retired, and her heir Walker continues to be supportive of our efforts. And of course, these companies, Lexio Therapeutics, Solid Biosciences, Tenaya, and Thrive Therapeutics, all of whom are working in this SADS space, working on new treatments to really have personalized medicine. And Guido has been hard at work promoting our story, and now we're on Apple Podcasts. I know I've shown you Spotify and other outlets where you can get this. So now we're on Apple Podcasts. So already starting to feel a bit like back to school. And back to school, of course, is an important time for our kids. And so many of these SADS conditions do affect young people, high school kids, even younger, infants sometimes. And so we always like to participate in a back to school safety. And there's lots of conversations going on about, is there a defibrillator at my school? And what does the teacher need to know about my medication? So it's a good time to renew our safety plans and the SADS awareness. Check out our website for that. Looking ahead to next month, we're going to have a familiar face, Dr. Andrew Cron. And he is well known to the not just the Canadian SADS community, but the international SADS community as a leader. And he certainly has done a tremendous amount of work. We're going to be talking about long QT syndrome next month. But today, very excited to have a good friend and incredibly gifted SADS researcher is Dr. Andrew Landstrom. So I'm going to stop sharing my slides here and bring him into the studio. It is always a pleasure to visit with him. So, Dr. Landstrom, Andrew, welcome. Thank you for making the time to join us here today.
Sure, thanks for having me.
So, I always like to have give our audience a bit of a chance to know who it is that we're talking with, and in this case, how we really are in the presence of greatness. And so a little bit of your background. How did you end up in cardiology and then in this genetic heart rhythm space? Take us through a bit of that.
Well, Shu, I think it might not surprise you, but I've always been kind of a nerd. I grew up loving science, loving the way in which science could impact our lives. I've loved genetics since I was little. And all through college, I was exploring ways that genetics could be used to help people. When I got to medical school, I thought I was going to be an oncologist because at the time, genetics was really becoming a way in which cancer therapies were being redefined. And I found out very quickly that oncology just was not the clinical space I wanted to be in long term. And so I had kind of a moment and didn't quite know what I wanted to do. At the time, I was up at Mayo Clinic as part of their MD PhD program, so training in both medical school and graduate school to be a scientist. And I did what most graduate students do, especially when they don't know where they want to be. You kind of rotate around. And so you get to do these little lab rotations, these little short stints with various scientists around until you find a lab that you really like. And I did a couple of genetics rotations in people that do something called molecular pharmacology, right? How genetics can influence therapies. And finally, I found my way to a lab, headed by a guy named Mike Ackerman. And I didn't know it at the time, but that was a pivotal point for me. Of course, Mike is very well known to the SADS community and leads the SADS US cause. But I didn't know that at the time. I just simply loved how genetics was being applied to heart conditions, many of which are SADS conditions. And I fell in love. I then went back to medical school and fell in love with pediatric cardiology. From there, I went down south to Texas Children's Hospital for additional training in pediatrics and pediatric cardiology, and I did a little extra research training down there too to learn more about how heart cells at a very basic level generate electricity and how that can go wrong in arrhythmias and SADS conditions. Then had the chance to come back home to North Carolina. My wife and I met in North Carolina as undergrads at a different university down the street from Duke, where I work now. That university has a slightly different shade of blue and a slightly better basketball team. But we had a chance to come back and come back to North Carolina that we love and raise our kids. And since about 2018, I've been back in Durham, working at Duke. And I get to do what I love. So I get to see patients with SADS conditions. I'm a pediatrician, so my patients tend to be a bit younger, much like yours. I get to see young adults as well, and sometimes whole families. And then when I'm not seeing patients, I get to study how SADS conditions develop. And a key goal of that research is to try to figure out new therapies that might be more precise, more effective than the therapies we're using now, and potentially to figure out how the disease will progress and to predict disease risk before it ever happens, which I think would be the holy grail. Because if you compare, prediction with great therapies, you might be able to change SADS conditions as we know it.
That's a wonderful journey. And of course, our audience recognizes Mike Ackerman, and I'm sure the chemistry in those early days must have been great, the pun intended, but because of course you both have contributed so much. And now you're also an editor of one of the circulation journals. Just to give yourself a little plug there, so our audience really understands here who we have in the studio today.
Well, I do serve as one of the associate editors for a journal called Circulation, Genomic and Precision Medicine, which is, in my opinion, kind of the go-to place in the scientific literature for research studies that involve genetics and precision therapies. And it's really a labor of love. So I get to see the science that's being submitted to journals from scientists like you, Shu. I get to read the science sort of before anyone else does. And the best science, it's our pleasure to publish it and to serve as sort of a medium that connects scientists with other scientists and through many people in the SADS community. Circulation, Genomic, and Precision Medicine is one of many journals, of course, where scientists publish their research, and it's for the benefit of the scientific community and the benefit of the whole SADS community. And so you can actually go online right now, find the journal, and actually look at the science yourself. And a lot of it's tremendously exciting. It's an exciting time to be a scientist, to be a physician who cares for patients with SADS conditions because of all these new discoveries that are coming down the pipe.
And I mentioned the editor position very intentionally. I think it's very important for our viewers to realize that before before something is going to come out in a published way, ready for sharing, it's gone through a lot of rigor and scrutiny and peer review, and some of the journals now, or some of the submissions, are actually available before that peer review process. So one has to be a little bit careful about, it's not fake news, but it's sort of unedited news. So it's, you're always best off getting your information from something that has already gone through those rigors. So speaking of breakthroughs, we're talking today a bit about gene therapy, and we've talked a bit about that on this show before. But I always, I think it's such a, it's such a fascinating topic, and I think that there's a lot of progress, but there's also a need to remind our viewers. And as you said, a lot of the genetic discovery occurred in cancer, in that field, because you had abnormal cells that were really multiplying, and there were genetic abnormalities. So the cardiac genetics that we're talking about, particularly in these SADS conditions, is quite different. So let's just let's go back for our viewers here and for ourselves. What does a gene have to do with SADS conditions?
So I sort of think of genes as the chapters in a book, and the book is the book of all of us. So our DNA, who we are, is defined by A's, T's, G's, and C's in our genome. And that's our instruction book of how to make you, how to make me, how to make all of us. And each of those instructions kind of come in little packets, little chapters, and each of those chapters can be considered a gene. In our heart, many of those genes are instructions for making very important components of our heart cells. Those heart cell components are needed for our heart to beat, for our heart to move electricity appropriately, and then for our heart to contract. Now, sometimes just by the nature of copying things over and over, there can be little changes that are made. And there are changes that are made when we have kids. That's part of the reason why we're all a little bit distinct genetically. Sometimes those changes, which occur randomly, can occur in genes in these chapters that make important proteins for the heart. And when that occurs, sometimes those changes can lead to abnormalities of the heart. And so many SADS conditions have as their underlying cause these types of differences in DNA. And so, 15, 20, 25 years ago, the goal was to get everybody sequenced so that we could identify what that cause was, that genetic misspelling, because that's informative for what the diagnosis is, what your SADS condition is, and how best to treat it. But now, Shu, kind of as you were saying, it's an exciting time because of all these rapid advancements in things like gene therapy and precision therapies. Now we can take a step beyond just what is the disease, and those same mutations that cause disease are now the same things that we need to know about to treat the disease more effectively with some of these gene therapies and precision therapies.
I've heard the analogy of the book, and just for our audience, the book has about 20,000 chapters. If we think about genes being a chapter in a book, and then you can have spelling mistakes that don't matter, or that are just don't change the meaning, and some of them suddenly the chapter doesn't make sense anymore, and it ends right in the middle of the cliffhanger. So great analogy. So let's talk about the process of gene therapy, because as we know, our DNA is built into our cells, and the DNA in our hearts is the same as the DNA in our muscle, other muscles. So how are the companies, how are the researchers approaching getting these corrective therapies where they need to be?
that's kind of the $1,000,000 question, is how to do it correctly, how to do it safely for all the different genetic changes that can occur in a SADS condition. Sort of taking a step back, it's amazingly complex. So, a SADS condition, we'll just take long QT, can be caused by those misspellings in lots of different chapters. So you can have a misspelling in one gene, say KCNQ1, the cause of long QT syndrome type 1. You can also have a misspelling in another gene, like KCNH2, and still have long QT. In addition, patients with one type of long QT, like KCNH2 mediated or Q1 mediated, long QT 1 or 2, can have lots of different mutations that lead to the same type of long QT, even within the same gene. And so the gene therapies then, to be able to reach as many people as possible, have to be very creative. They have to be able to account for all the different misspellings, all the different mutations that can get you to a particular kind of disease. And then the way in which those misspellings cause disease can be very different from chapter to chapter, from gene to gene. In sort of the simplest scenario, the misspelling causes the gene to not make any more sense. It's sort of like you were saying, kind of that one single change that throws off the whole cliffhanger. You have no idea how the story ends. Might as well just have not even read the chapter. In those scenarios, we call those loss of function mutations, where those mutations lead to just the chapter doesn't work anymore. You don't even bother reading it. And so there's no protein, no product that's made after that mutation comes into play. And so the process for fixing that from a gene therapy perspective, in theory, is relatively straightforward. All you have to do is put a normal chapter back in the book. And so that's called gene replacement. That has historically been the earliest way in which people have tried gene therapy. That might work for some SADS conditions. There are other challenges, but that's sort of the simplest thing. I think it's a bit more complex from there, because sometimes the mutation, the misspelling in that chapter, doesn't just cause the chapter to make sense. It actually causes that chapter to not make sense, and maybe other chapters to not make sense too. And so simply replacing the chapter doesn't always work. And that's where newer strategies are needed. And it's an exciting time because there are newer technologies that can be used to try to get clever, to try to figure out ways around this complexity.
And in general, we're not going in and changing people's DNA. So we're not able to change that spelling mistake and then have the DNA start working properly. We need to work around it, either by providing a healthy copy of the gene or working with the protein. So let's talk about a condition that where we're seeing some trials beginning. So arrhythmogenic cardiomyopathy, ARVC, for example. So people are starting to use some gene therapy trials, early days, I believe. Why aren't we just getting everybody in the ARVC clinic, lining them up, roll up your sleeves. Here's a shot of, new plakophilin.
Yeah. I would love if that's the case. I hope that will be the case down the road, but you're right. For now, it's a very specific study that's ongoing. And that study, and actually there's multiple companies that are interested in doing this, but the principle is all the same. As you mentioned, it's not just any kind of arrhythmic cardiomyopathy. So if you have arrhythmic cardiomyopathy, it's good to know your gene mutation because it matters. And this is a perfect situation of where knowing that information matters. Mutations in one of those genes, plakophilin, PKP2, is one of the more common forms of arrhythmic cardiomyopathy, one of the most common causes. And these mutations cause that loss of function, meaning the gene just doesn't work. And so if you have that mutation and the gene doesn't work, then this gene therapy, in theory, might work well for you. That gene therapy uses a clever delivery tool because DNA doesn't really want to go into the heart. You can't just, To extend our analogy, put a chapter back in the book by itself, you need a vehicle to return that book chapter in. And it uses kind of a clever one, which is a scientifically created virus called adeno-associated virus. And viruses love to put DNA into cells, and you can use this same trick to deliver a normal copy of PKP2 to the heart. And so it's really early days for this. This adeno-associated virus, this delivery tool, has shown to be effective in delivering things to the heart. But like all viruses, the immune system can react to it. And normally when an immune system, like our immune system, reacts to a cold or a virus that we may come into contact with, sometimes we can get a fever, get a cough. Sometimes we can get really quite sick. Just like in that scenario, sometimes people who get a virus for gene therapy can get really quite sick because of it. And if we're giving a lot of virus, like a ton of virus to be able to deliver gene therapy, you can get very, very sick. And so those are one of the things just to think about. And the scientists and the FDA and the people running the trials are very, very concerned about moving forward. And so we're very early days. We think of new therapies as requiring multiple stages, multiple phases for the approval process to make sure it's safe and that it works. And this is the earliest. This is a phase 1, meaning it's all about safety. And so it's early days, but if you have that specific gene and you meet criteria, you might be eligible for an early phase study for safety. The hope would be that if it is proven safe, that the follow-up studies, the phases that come after that, would expand the number of people who might be eligible, continue to look for safety, look for evidence that the therapy is working. Just because something works in theory doesn't mean it works in practice. And that process also takes time. But my hope would be that if it is safe and shown to be effective, that we may have a gene therapy down the road. And that can be 5, 10, 15 years from now. But that will be an important thing for us going forward. And then my hope would be that the other genes and people who are working on those other genes come up with, again, clever therapies that can do the same for other types of arrhythmic cardiomyopathies and other forms of SADS conditions. But we're a little ways from all of that right now.
Yeah, I think, such important points that this is, relatively early in the science, but the fact that we are, that we've engaged and are talking to patients is really exciting. So we've come fairly far. Some people who have done their research know that the early trials of gene therapy were really complicated by the fact that you had to suppress the immune system to let this new DNA, these new proteins be accepted. And so that, that's not without risk. So exciting that there are patient conversations happening. And I feel in general that the companies are being very thoughtful about this. They're working closely with clinicians like us and talking to us, of course, to also, and they're also talking to patient groups, and the SADS organizations are at the table of those conversations. So we're both pediatric specialists primarily. So, the early waves are mostly adult patients. What sort of patients do you think are well suited for these early gene trials? Are the companies looking for, the healthiest of the volunteers, or the most, the sickest, the most severe cases? How are you looking at your population and what's going on?
Yeah, it's sort of a middle ground. I think if people are really quite sick from their SADS condition, kind of running out of medical options, and you're very sick because of it, sometimes experimental therapies, especially in gene therapy, may not be always the best option. Conversely, if your SADS condition is well managed on the therapies that we have available, and you're feeling good and you're able to live your life, then these are early days for these studies. So maybe taking part in an early phase trial like this where the outcome is it safe, may not be the best option for you. I think a lot of the companies, and I think you're right, they are being thoughtful about this, that are doing trials, are looking for the kind of a Goldilocks phenomenon where the porridge is not too hot, not too cold. People with SADS conditions that are serious, not incredibly, extreme and quite life-threatening, but also, not people who are well managed. Those are the patients that oftentimes kind of are in that Goldilocks, right in between two extremes, where they might find something like that worthwhile. But I think beyond kind of what the companies are looking for, I think these families themselves are just a specific kind of family. As you mentioned, it's early days. These are kind of like the explorers. These are the patients that say, we're doing this not necessarily because we want this to be a therapy for me, for my family, for us, but rather we want to help pave the way for families to come after us, because it is early days. We don't know the full safety profile of these new gene therapies that are being tested. And there's a lot of unknowns. And so they're kind of the explorer among us, right? Who say, we want this for others that will come after. And so it's a very specific kind of person that, is taking part in these early trials. And they're individuals who have made that determination for themselves.
And as you allude to, many of the protocols really require quite a close relationship to the hospital that's doing the treatment. So you're there, in most cases months, with fairly close relationship. So it's not necessarily at a stage where it's widely accessible, and I think that's completely appropriate, because not every facility can do this therapy. I picked a sort of a tricky example. Arrhythmogenic cardiomyopathy, ARVC, is usually progressive, and so we don't necessarily think that we're going to be able to reverse the process. We don't know, the duration of the effect. Another condition that you and I both spend a lot of time and thought on is CPVT, catecholaminergic polymorphic ventricular tachycardia. And in that case, I think it's quite different, because these are, primarily young people, but there's not necessarily a progressive nature to it. So the gene therapy then starts looking very different. And I don't know if you've had any patients or any involvement with some of the CPVT gene therapy discussions that are also ongoing.
Yeah, I think CPVT can be quite challenging to treat. I think it's one of those diseases where we have a good cocktail of medications and left cardiac sympathetic denervation surgery, which in the right combination can be very effective. But certainly there are patients that, despite those therapies, still have challenges. And yes, there are some gene therapies that are being considered and some new precision therapies that are being considered. Again, early phase trials, the early days safety studies for those. And my hope is that, just like with arrhythmic cardiomyopathy and PKP2 gene replacement, that maybe in 10, 15 years from now, we'll have a lot of those trials show safety and show that the medications work, probably alongside the other medications we currently have. And that would be great, because I think we need more options for patients with CPVT for sure.
Yeah, and you mentioned an important point there, that especially these early trials, and by and large, they will keep all the existing treatments in place. So I know lots of our patients and lots of our viewers have, for example, beta blockers, and so the science has not reached the point where people can have gene therapy and then come off their beta blocker. We have not reached that threshold yet. That is, I'm sure that's the goal of many people, is to have sort of a single treatment. But the science has not arrived there. And so back to Dr. Landstrom's role as an associate editor of a very important journal about this very subject. We have some work ahead. What are some of your what are some of your learnings over this past 5 years where we've been talking about the gene therapy a lot more? What are some of the things you're excited about? What are you seeing that concerns you? You've put a lot of thought into this.
Well, I'm very excited about the creativity of a lot of the science that we're seeing. So, there are creative ways that are being put forward as potential new gene and precision therapies for nearly every SADS condition. And there are challenges with nearly every SADS condition. And somewhere, some scientist is thinking up ways to go around, to go over, to go under those obstacles. And there is a lot in the literature now that gives me a lot of reason to be excited. A lot of these, we call them preclinical studies, so things before they reach a clinical trial. That space is rich. And I would say probably we're at a watershed moment, meaning the floodgates are opening and we're seeing tons of this research because the tools scientifically are so good. And so it makes me excited about what might happen and when many of those preclinical studies become clinical trials. I think we will see in the next 2 decades a complete sea change in how we start to treat these diseases. I really think so. Now, we can debate about what therapies, under what circumstances, but I think we're going to look back in 20 years and say this was the time where things started to change. But with everything that's exciting, there's always a little bit of risk. As more and more people have gotten things like gene therapy, we've become more aware of the problems with gene therapy. We've talked a little bit about the virus, the problem with the immune system. That is still a problem. And that's something just to know about. And we need to address collectively as scientists and as clinicians if we're going to get gene therapy right. It may be that we leave behind viruses. It may be that we find other delivery mechanisms. It may be that we do things to prevent the immune system from attacking the virus or vice versa. So there's lots of ways around it, but it's just something we need to be aware of. And then, I think families, just need to stay informed. You're doing a great job by logging in and joining the SADS community, and Canadian SADS and US-based SADS do a great job of engaging the SADS community. And I think we need to keep up the conversation. I think families need to know about their SADS condition, be informed, and be informed about the science that's happening around their SADS condition. Because then, when these new things come to those early phase trials or maybe later phase trials, you're already aware. You know all about it. You know your family's mutation. You know whether you may or may not benefit from a precision or gene therapy, and you can make an informed decision for you and your family. And that's really exciting too. So I think it's an exciting time, and we just need to continue to have conversations about what it means for the SADS community.
Yeah, wonderful overview. I'll ask you a couple of hard questions here. So lots of our families, of course, they don't have a genetic marker. So Brugada Syndrome, for example, where really only the minority of patients have a genetic explanation. There are definitely genetic components, but not sort of a single gene. Can you foresee a time where even some of these, genetically elusive conditions like Brugada could benefit from gene therapy or other sort of replacement therapies?
Absolutely. So, I think the unifying thing behind something like Brugada, even if you don't have a gene defect, is that Brugada kind of manifests the same way electrically in the hearts of everybody. Some of those people have a mutation in the sodium channel. Other people simply don't. It doesn't mean the disease is fundamentally different. And so my hope would be that over time, we could then learn how to attack the fundamental process of the disease, not just the SCN5A sodium channel gene variants in some. And I think you could make that argument for every SADS condition. Can we actually alter the fundamentals of the disease, not just target the specific gene? That science is a little bit further behind, I will say, overall, but the opportunity still exists. And there is some really great science going on right now to try to think around a specific gene and think about a specific disease more generally. And my hope would be that we continue to see that science progress so that we can have trials, and we'll know it when we see it. A trial that says we'll take everybody with arrhythmic cardiomyopathy. regardless of your gene. We'll take everybody with Brugada, regardless of your gene. And that'll be an exciting day for sure.
And I think the SADS conditions lend themselves so well to studying these arrhythmias, because most arrhythmias are occurring not in our SADS population, but rather in older people who have advanced heart disease, congestive heart failure, have had heart attacks, those kind of things. But those are very complicated models. There's not necessarily one cause or one protein involved, one gene. So the SADS conditions really allow researchers and companies to be very mechanistic, and this term that keeps coming up, precision medicine. So we're not going to treat every heart protein. We're going to really narrow in on the problem. So Andrew, in the last few minutes here, I'm gonna ask you if there's anything that you in your lab have been working on, because I know I've had the privilege. You have a wonderful group of young scientists and up and coming researchers, and I know you've done some really interesting discovery work on ultra rare causes of arrhythmias. Is there something that you're working on now that we can let this audience have a little sneak taste of?
We are always interested in trying to make those new discoveries that might inform new therapies. We have had kind of a nice discovery of late, and we found a new genetic cause of arrhythmic cardiomyopathy in a gene that many people might not have heard about. That gene is called TAX1BP3, TAX1BP3. And we think there's probably only a handful of families around the world who have the kinds of mutations in this gene that you need to get really bad arrhythmic cardiomyopathy. And so ultra rare. But we spent a lot of time trying to figure out why it is that they got disease in the first place. By and large, the people with mutations in this gene get very severe arrhythmic cardiomyopathy much earlier than most patients do. We're talking childhood years, which is how I stumbled upon a family with it. And if that's the case, then kind of going back to that precision medicine, could we use this ultra rare family to learn a lot more about how the disease progresses more generally? And we found a particular ion channel, a particular sort of product from one of those chapters in the book gets made by the heart cells as they're struggling with this mutation. And if we block it, so if we use medicines to block that ion channel from having its effect, we have a really nice way of actually suppressing the arrhythmias from the heart cells with this defect. And when we try that same approach with other patients' heart cells, with other mutations and other types of arrhythmic cardiomyopathy, it seems to be working there too. And so this may be one example, we think, of where an ultra rare family has given us a glimpse into maybe a new therapy down the road for arrhythmic cardiomyopathy that might benefit everybody, regardless of genotype. And that's particularly exciting. So we're very excited about that. And you'll have to have us back in a little bit to keep you up to date as to how that search is going.
Well, I knew I could put you on the spot because I've followed your science closely from the early days when you were recognized with awards at the Pediatric Electrophysiology Society. So, Andrew, it's been an absolute delight talking to you today. I always learn so much, and you have such a nice view over really what's a very complicated field that is, it's starting to come into our daily conversations a lot more. So I'm glad to have a partner like you working on these things.
Thank you, Shu. Pleasure is mine.
Well, thank you so much for joining in. Dr. Landstrom is modest. He's really contributed a tremendous amount to the field, and we in the Canadian SADS Foundation and the SADS organizations all over the world are fortunate that we have these researchers working hard on on insight and cures. So that's it for our not quite our last show of the summer, but next month we're going to have Dr. Andrew Cron, and I hope you have a good last few weeks before it's back to school. Remember to check out the website, Canadian SADS, the SADS.org, SADS.ca, and check out our back to school information there. Goodbye.